Okay, so here we are at concept 11.4. We're on the last stage of signal transduction pathway. We got to get a response. So after all of this, after the reception of the signal and the transduction of the signal and all these various chemicals flying around inside the cell, what is the final change or output that we're looking for? So 11.4 says response. Cell signaling leads to regulation of transcription or cytoplasmic activities. So usually we're either going to be causing a change in transcription, which is basically going to the DNA and saying, okay, make some messenger RNA, some instructions to tell the ribosomes to build this particular protein, or kick on this enzyme, or turn off this enzyme, or cytoplasmic activities. And those would be anything else that's going on inside the cell. So what happens when the cell receives a signal and it goes through this whole transduction pathway? Well, we get to our final response, oftentimes called the output response. Ultimately, our initial signal, the signal transduction pathway, is eventually going to lead to some sort of regulation or change in one or more of the cell's activities. Sometimes this happens in the cytoplasm and in all the various organelles or parts of the cytoplasm. Sometimes this actually happens within inside the nucleus. Many times the signaling pathway changes or regulates the creation of a particular enzyme or a protein. And it usually does this by turning particular genes on and off within the nucleus, and then that causes particular strands of messenger RNA to be sent out to tell the ribosomes what to do. The final activated molecule in the signaling pathway may function as a transcription factor, which is exactly what I just said. Synthesizes messenger RNA, which is basically just the blueprint that the ribosomes follow to create their proteins. So here's a quick model of how this occurs. And of course, this is very compressed. We're trying to clean up all that yucky middle part there. So let's say the signal molecule is some sort of growth factor that changes the way the cell grows or reproduces. We have the receptor molecule here. And then we have, of course, that messy middle part, transduction. We have a whole phosphorylation cascade. That's going to cause some particular protein to enter inside the nucleus. It will activate this transcription factor. The transcription factor is just something that affects transcription, which is taking the instructions from DNA and creating messenger RNA from them. Then the response is to basically code a piece of the DNA as on or off, create from that messenger RNA, which then will go to the ribosomes to be built into proteins. Back up there. Yep. So transcription, or turning on or off portions of the genetic code to then send out messenger RNA to the ribosomes, oftentimes is the end goal of this whole entire signal transduction process. Another good example of what can be the final end product of this is the regulation of particular enzymes. So rather than building new protein enzymes, sometimes we're just slowing down or speeding up ones that already exist. So we can regulate the activity of enzymes rather than create new ones. Sometimes that's our end goal. A great example of this is that fight or flight mechanism where epinephrine is the signal that leads to the breakdown of glycogen into glucose, and that occurs usually inside of the liver. So glycogen is that short-term storage inside the liver that the body can break down very quickly and use as a burst of energy. And so that is a very good example of enzymes inside the liver basically being turned on or allowed to go quickly to break down that glycogen and to allow for the fight or flight system or response that we see. What's that? Did you say that you wanted to know all the steps involved in the breakdown of glycogen based on epinephrine? Well, don't panic, because of course you don't have to know all this. There's no way I would make you memorize all this. This is just an example. You do not have to know this at all, but this is just an example of all the steps. So this is reception. This is the binding of epinephrine to a particular G protein coupled receptor, and then there's this whole cascading process. This is all transduction. We end up with basically a response where a particular enzyme is breaking glycogen down into glucose, and that can be quickly used for energy to either fight or run to hopefully live another day. So don't panic. You don't need to know all this. This is just an example of the process that occurs. So oftentimes these signaling pathways affect one small portion of the cell or one particular activity, but sometimes they actually affect the overall cell, the overall behavior of the cell. And this is especially true in microorganisms that have only a few cells, or in single cell organisms. And a good example is the change in cell shape, say for example in the yeast cells that we were talking about. So remember, we mentioned that there's an alpha model and a beta model. They're not really male and female, of course, but there's different varieties of yeast cells, and they want to try to combine their genetics to strengthen basically their genetic pool. So the way they do that is they actually have a signaling mechanism that affects their growth of the entire cell, and they will grow toward one another. They're not very motile. In other words, they can't move around very easily. They don't really, obviously, they don't have limbs, feet, and things like that, and they don't even really have pseudopodia or flagella or anything else. So how do they get together? Well, they actually will grow together to mate. And the way they do this is because of the various signal molecules that are sent out by the opposite variety. It causes a change in the entire cell, and they grow what's called a shmoo. And it's called a shmoo because there was apparently a cartoon character in the 50s. I wasn't around in the 1950s, but apparently there was a cartoon character in the 1950s that kind of had this shape. Here it is right here, and he was called shmoo. And when I show you the yeast cells, you're going to say, yeah, it kind of looks like a shmoo. So here's the process. We don't have to know all these particular details. So again, don't try to memorize all these details, but just take a look at the overall process. We have this mating factor. Remember, that's like a pheromone that was sent out by the alpha variety or the beta variety, and it attaches to a receptor protein in the opposite variety's outside cell membrane. And this is a G protein coupled receptor, so then it attaches to a G protein. GDP is created into GTP, which activates it, and then we go through the whole cascade process. And eventually what this creates is it changes the microfilaments. Remember, microfilaments are part of the cellular structure network, and it grows microfilaments. And so this basically causes a projection, a growth to form in this particular portion of the cell, and that growth is called a shmoo. And you can kind of see the picture up here. I'll show you a bigger picture in just a moment. So here we have yeast cells, which generally are pretty much round, and they've dyed this in a way that shows the most actively growing parts as red. So you see these little projections sticking out right here. These are the shmoos, and this is the growth of the yeast cell as they grow toward one another. So because they can't walk around or swim around, they grow toward one another, and that's a complete change in cell growth caused by this particular signal from the other variety. So if you look at them, they are kind of cute. They do kind of look like a shmoo. So there he is right there, shmoo. The other thing we mentioned is because this is a multi-step process, the response is often not just on or off. Signal transduction is usually not just black or white and on and off kind of affair. There's a lot of different aspects to it, and there can be some fine-tuning along the way. So because that there are many steps to the process, you've kind of got four things that can happen to kind of fine-tune the process. One of those is amplification of the signal. So one signal molecule can then kind of be amplified into numerous signal molecules that can cause a bigger amount of change. One of them is the specificity of the response. A third one is the overall efficiency of the response, and we'll take a look at that. And then the last one is the termination of the signal. So let's kind of talk real briefly about these four things and why sometimes the confusing multi-step process of cell transduction, cell communication, can actually be a good thing in some ways. The first one is signal amplification, and that one's kind of easy to understand. We can have a single signal molecule, epinephrine or a hormone or something like that, that can cause a cascade of changes, and this cascade can then affect a wide variety down here at the bottom. So like in this example, you have one particular molecule that is affecting a G protein receptor, and then that gets amplified and it affects a variety of adenylate cyclase enzymes. It gets further amplified by the CAMP, and then now you have all these different kinase proteins down here, enzymes able to do their job because of this one signal molecule. So amplification allows for a huge number of activated enzymes or activated products to come about from one single molecule. The other thing that having multiple steps allows for is specificity. In other words, we already mentioned how not every cell is activated, or not every cell is affected by every signal um, molecule. Well, as a matter of fact, different cells can be affected in different ways, and that's because different cells turn on different sets of genes. So a skin cell looks like a skin cell and acts like a skin cell, and it looks different from a muscle cell and acts different from a muscle cell because different genes are turned on inside the skin cell. So when you have, say, for example, epinephrine, when that particular signal molecule gets to a skin cell, it may have an activity that it causes, but that activity is likely to be different from the activity that it causes in a muscle cell. So specificity of the signal and then specificity of the resulting product or process is allowed because of multiple steps within the whole process. So because there's so many different proteins and steps involved, that allows the cells to detect and respond to different signals. And then it allows them to kind of respond to that signal in a different way based on what type of cell they are. So again, another good example I wrote down here is heart cells and liver cells don't react the same way to epinephrine. When heart cells get the signal of epinephrine, it causes them to speed up and beat faster and harder because heart cells are a type of muscle cell. But liver cells don't do that. Liver cells start the breakdown of glycogen when they receive epinephrine. So the same chemical can cause different reactions in different cells, and that's because you have this fairly complex process going on. That also allows for branching and what we call crosstalk, where you have this kind of a network of communication going on between cells that are alike, like, say, all the liver cells are kind of talking to each other in a way, but then you also have communication that goes across the entire body between heart cells and liver cells and different organs in the body because of these pathways. So here are some of those examples of different types of specificity or changes that can happen. Here's our basic example where a signaling molecule comes in, attaches to the receptor, then you have relay molecules, transduction, and then you get a response. So this is kind of a linear, you start here and you end up here. The second example here, you have the same signal molecule comes in, and that goes to this particular relay molecule, but then that activates two different other molecules, so we end up having two different responses. So the one signal causes two different responses. In this activity here, this third one, we have two different signal molecules coming in, and they kind of interconnect. This one has this pathway that could end in response 4, but this one can either activate or inhibit this. So depending on what signal this molecule is, that can slow this process down, or it can help speed the process up. So the combination of these two signals kind of affects response number 4. That's kind of a version of crosstalk. And then the last one here is this signal is the exact same as this one, but notice the receptor is different. So that causes a different signal pathway, and we get a different response. So the same signal, even within the same cell, but different receptors, can cause a different pathway and a different response. The third part of the response we want to look at is what's called scaffolding proteins and signaling complexes. Again, this seems like, and you would think this is not a very efficient process, that it doesn't seem like there's an easy way to get these molecules from one place to another and to get them all into the cell and to get the whole process kicked off and going. It's not a very efficient process. So the cell does have a couple of ways to make it a bit more efficient, and one of those is by using what we call scaffolding proteins. Scaffolding proteins are very large proteins, relay proteins, and they have other relay proteins attached to them. So think of them kind of as tour buses or tour guides, and they are carrying other receptor molecules with them to kind of put them all in one place to kind of make it easier for this whole process to occur. So scaffolding proteins can kind of make this whole process more efficient because they bring together all of the signaling molecules involved in a single pathway. So if you have, say, 2 or 3 people that all need to work on one particular job, and if you were to have them carpool together in the mornings and in the afternoons, that might help get the job accomplished because you put those people together to get the goal accomplished. They're in closer proximity to one another, and hopefully the final job can get done a lot quicker. So in some cases, these scaffolding proteins also help activate the relay proteins by turning them on, turning them off, things like that. Let's take a look at a picture, which is always better. So here you have a signaling molecule and the particular receptor. And let's say there's 3 different protein kinases that are going to be involved in this whole process. Well, if you have a scaffolding protein that can attach to the receptor and can kind of carry all 3 of these protein kinases, well, now you've got them all grouped together, and they're already all together where they need to be, so the whole process can go a lot faster. Otherwise, you have these 3 protein kinases just floating all by themselves out here, and it may take a longer period of time or a greater amount of chance for everything to occur. But the scaffolding protein kind of facilitates this whole process, kind of says, hey, okay, these 3 proteins are going to be involved. If they were all 3 together, then that whole process would occur faster. Now, I say that knowing that, of course, proteins aren't thinking like that. That's not how biology and evolution works, but I'm just kind of humanizing the process to make you understand, to give you an analogy. Finally, the termination of the signal. What do we do when we're all done here? This signal, kind of like a jolt of chemical activity. It can't go on forever. When you get that epinephrine that floods your body, if you're riding a roller coaster and you get that jolt of epinephrine and your heart beats faster and you start to sweat and your stomach gets a little queasy, some people like that feeling, and it feels good when you ride a roller coaster and you're excited if you know you're safe. But you wouldn't want that process to go on for the next 2 hours. And just like that cheetah and that gazelle at the very beginning of our chapter, that fast beating of the heart and quick muscle activity, that can be only good in the short term. You don't want that switch to stay turned on, or that's going to cause some damage. So we need some way to inactivate this mechanism. And the inactivation of the mechanism is a very important part of cell signaling. It does you no good to have an on-off switch if it's stuck in the on position or stuck in the off position. It has to be able to be reset back to its original state. So usually what happens is when the concentration of signal molecules starts to fall, fewer of the receptors will be bound and activated, and you'll have the ability to kind of reset back to the initial state. So after the excitement is over, after the cheetah has caught his meal and eaten his breakfast, or perhaps after the gazelle has gotten away, the epinephrine within the body, the amount of epinephrine is lowered, and then the whole process can go back to its original off state. Let's take one last pause right there. We're going to look at one last cool thing called apoptosis. Apoptosis is a particular process involved in cell signaling that's kind of very interesting, very cool. Apoptosis is basically programmed cell death, or predetermined cell suicide. So we'll look at that in its own little chapter in just 1 second, or its own little video in just 1 second. So take a break, come back for apoptosis.